Howdy, my name is Jazlyn Gonzalez. I am part of group number 6 with Andres Garza, and today we will be presenting over our biochemical test part 2. Biochemical tests are essential tools in microbiology used to identify and differentiate bacterial species based on their metabolic characteristics. These tests detect specific enzymatic activities or metabolic products that reflect how microorganisms utilize or break down various substrates. In this second part of the biochemical testing series, we focus on amino acid decarboxylation, particularly through the lysine decarboxylase and ornithine decarboxylase tests. These assays help determine a bacterium's ability to remove the carbonyl group from amino acids, producing alkaline amines that cause a detectable pH shift in the medium. By comparing known bacteria, such as Escherichia coli, Proteus mirabilis, Morganella morgani, and Salmonella typhimurium to an unknown sample, we were able to observe distinct metabolic patterns that aid in bacterial identification. By understanding these reactions, we can accurately classify and guide appropriate treatment options. Now for our materials. For media, we used LIA and ornithine broth. For cultures, we use Citrobacter frundi, Enterobacter cloacae, Escherichia coli, Morganella morgani, Proteus mirabilis, Pseudomonas aeruginosa, and Salmonella typhimurium. For equipment, we used a butane tank as our aseptic key source, an inoculating needle, an inoculating loop, and a tube rack. For our procedure of the lysine decarboxylase test, we made sure that the butt of the tube was stabbed, followed by streaking the slat with its respective culture. The tubes were then incubated and viewed for results. For the procedure of our ornithine decarboxylase test, we made sure the broths were carefully inoculated using an inoculating loop with its respective cultures. The tubes were then incubated and viewed for results.
Moving on to results and observations for lysine decarboxylase tests, we have several of the tests, which is with E. coli, M. morgani, M. mirabilis, S. typhimurium, and unknown 276. Starting off with E. coli, we have a positive test result, and this test result is able to decarboxylate lysine and produce an alkaline end product. As for S. morgani, we have a negative test result for lysine decarboxylase, and this is due to a slightly darker shade of purple and a hint of yellow at the bottom. As for P. mirabilis, we have a negative test result as well. This one, there is a slightly lighter shade of purple and a hint of yellow at the top. For S. typhimurium, we have a positive test result. This one is able to decarboxylate lysine and produce an alkaline end product. And lastly, for unknown 276, we have a positive test result. So this one as well is able to decarboxylate lysine and produce an alkaline end product. Now for the results and observations with ornithine decarboxylase tests, we have them with E. coli, C. fruindi, and unknown 276. For E. coli, we have a negative test result, and for the observations, we can see that there is a yellow coloration which reflects an acidic pH. For C. fruindi, we also have similar results as well, with a negative test result showing yellow coloration, which also reflects an acidic pH. And lastly, for unknown 276, we have a positive test result with a purple coloration, which reflects the basic amine putrescine being produced, raising the pH values. So in conclusion, the lysine ornithine decarboxylase test successfully demonstrated how amino acid decarboxylation can be used to differentiate gram-negative bacteria based on their enzyme activity. Positive results shown by the appearance of purple coloration indicated the production of alkaline amines from lysine or ornithine, while negative test results produced a yellow hue due to acid formation. E. coli and S. typhimurium tested positive for lysine decarboxylase, while M. morgani and P. mirabilis showed a negative or weak reactions, highlighting variations in their metabolic pathways. Similarly to ornithine decarboxylase test confirmed that unknown gram-negative organism shared biochemical similarities with E. coli and C. fruindi through its positive reaction. Overall, this experiment reinforced the importance of biochemical testing in microbial identification by revealing distinct enzyme activity profiles that help classify bacteria more precisely and support accurate diagnosis and treatment decisions. Next, we have our references provided for which we got our information. And this concludes our video presentation for our lab report. Thank you all for listening.